So
Good afternoon, everyone. I see people starting to filter in. I want to welcome you to our virtual Dementia Journal Club, broadcasting here from Winnipeg, Manitoba, Canada. We'll begin in just a few seconds. I see people starting to filter in. I'd like to ask everyone to mute their mics so that we don't have noise in the background. I'm assuming people can hear me and see me okay. We're going to turn it over to Dr. Guha in just a few seconds.
Yeah, I didn't realize there was a waiting room. Like there were a bunch of people waiting and I had to admit all.
Okay.
Good morning.
I'm seeing some familiar faces pop up. I also see some familiar masks popping up.
So probably you're seeing the name Gail Johnson. She's my current PI.
Not yet.
She doesn't have any profile picture.
I don't see any name though.
Will be
Oh,
just
wait. Now I do, yeah.
Yeah.
Okay, we'll just wait about another 30 seconds and then I'll give an introduction. So again, welcome everyone for attending our Dementia Journal Club, our virtual broadcast from Winnipeg, Manitoba. And I have 12:01, so I think we'll get started. So really today, it's my pleasure to introduce Dr. Sanjib Guha. He was born and raised in Calcutta, India, and he did his undergraduate studies there. In 2008, he moved to California, where he completed his master's degree. And after that, he moved to Singapore, where he worked as a research associate at the NUS. I'm not sure what the NUS is. Maybe he'll explain that. Then he moved to Barcelona, where he did his PhD. And after that, completion of that, then he moved back to California, where he completed his first postdoc at the well-known Buck Institute. Now he's currently at the University of Rochester in upstate New York. And as a globetrotter, he did his higher studies from 3 different countries. He's lived in 7 different cities and can speak 5 languages. Very impressive. And today he wants to talk about how post-translational modifications of tau selectively impact neurodegeneration and causes mitochondrial dysfunction. So this work has recently been accepted in Molecular Neurodegeneration Journal, and a corresponding review was published in Molecular Neurobiology Journal. Of course, that's where I'm an editor-in-chief. So we will post these papers on the Manitoba Dementia Journal or a Manitoba Dementia website, which you can find by going to LinkedIn and going to my profile. So without further ado, welcome Dr. Guha.
Thank you, Dr. Albensi, for opportunity for me to speak here. And thank you everyone for joining in. So yeah, so NUS stands for National University of Singapore, where I was there for like an year or maybe less. I'm just checking whether any other people waiting in the room or not. I think no one yet. So with that, I can start sharing my screen. Okay, all set. So thank you again for joining in today. And as Dr. Albensi mentioned, like I'm a postdoc at University of Rochester, and I have 2 wonderful mentors to work with, Professor Keith Narke and Professor Gail Johnson. And I'm very pleased to share all my data for today's talk. Like as Dr. Albensi mentioned, like most of the stuff has already been accepted. So we are starting a new project, and some of the data I will show, and like if we get time, we will move into some of the future direction as well. So for the topic of today's talk is the role of Alzheimer's disease relevant tau modifications in neurodegeneration and mitochondrial dysfunction. So for the next 45 minutes or so, like I will be briefly touching on all of these different points, which is the outline of my talk. Like first example, like in our lab, our main research interest or the disease of interest is Alzheimer's disease. And we specifically work on the protein tau and also the different modifications of that tau protein. And we use the C. elegans as our main genetic model organism to phenocopy all the disease pathogenic phenotypes. And also when we go inside more into the cellular level, I will briefly mention like all the different biosensors that we use in our lab to mainly capture different mitochondrial activities. And as I said, like if we get time, we will like finish up with the future studies and also the current working model. So as all of you know, like Alzheimer's disease is a severe progressive neurodegenerative disorder, and it's also the most common form of dementia, affecting millions of people worldwide. And also it has been mainly seen in the aged population, like people over 65 years of age, they are mainly affected, and which causes them, the main clinical symptoms are decline of memory, loss of cognitive function, and their changes in behavior and plasticity. As very unfortunate so far, like there have been no therapeutic drugs that are present that can cure, totally 100% cure this disease. The two main pathological hallmarks, as we know, is the formation of extracellular plaques consisting of a beta peptide, and also the formation of neurofibrillary tangles made up of hyperphosphorated tau protein. So as I said, in our lab, we specifically focus our interest on the tau and the different modifications. So what it has been known so far, like there are soluble tau oligomers, which basically accumulates at the synapses of brain, blocks synaptic connection among the neurons, which eventually causes neurodegeneration and death of neurons, and eventually causing shrinkage of the brain as compared to the healthy brain and affecting all the nearly functions of the brain. But still, the main question is the mechanism behind such a beneficial protein or like a tau to become toxic and eventually causing neurodegeneration and neuronal cell death. The exact mechanism is still unknown, and that is what we are looking for. So tau, as we know, like is a microtubule associated protein. It's a natively unfolded. Structure-wise, it's like 50 to 75 kilodalton. It contains mainly 4 domains: N-terminal domain, foreign-rich domain, microtubule binding domain, and C-terminal domain. And it is encoded by the gene called MAPT, microtubule associated protein tau, and it's located on chromosome 17. And alternative splicing of that leads to 6 different isoforms based on the N-terminal inserts as well as repeat domains. So in our lab, we mainly focus on the 0N4R repeat out of all the 6 different isoforms, because there are a couple of reasons behind that, because that is the repeat that is mostly highly interact with the microtubules, and it has been shown that it causes more pronounced pathogenic events, as well as it is found in the adult human brain. So from now on, the 0N4R in my results will be written as tau T4. And as I mentioned, like tau in generally, like is a very beneficial protein, like all of us have tau, and it is mainly present in the axons, which helps in promotion of microtubule assembly, stabilization of the microtubules, which ultimately causes or develops neuronal development and polarity, axonal transport, and many other functions. But the problem happens like when it becomes pathogenic, it no longer stays in the axons where it gets missorted, which leads to the disintegration of microtubules because it cannot bind to the microtubule properly. There are a lot of free floating tau, which eventually causes neuronal disruption of neuronal communication, ultimately synaptic failure and neurodegeneration. So the question comes like how and why such a beneficial protein like tau becomes pathologic and causes all these different pathologic features. And very recently, it has been found out like there are several modifications, rather post-translational modifications, which actually causes the tau to change its structural functional integrity or the conformational change, which eventually leads to its toxicity. For example, when we like post-translational modifications, we call it in short like PTM. So the main different PTMs that we can see happening in the tau protein is phosphorylation, acetylation, ubiquitination, oxidation, etc. So when this kind of PTMs happens in the tau protein, it causes the changes of its structure, which eventually leads to toxicity of the tau protein. So among the different PTMs, the two main PTM that has been shown to be very pathologic in the human AD brain, first of all is the phosphorylation. That also happens early in the disease, like everything starts with phosphorylation. And again, tau being a phosphoprotein, it has like over 80 potential phosphorylation sites. And out of the different sites, the main pathological sites that has been found to be very critical is threonine 231. And next come after the phosphorylation is acetylation. That is also another major PTM that happens in the tau protein. And again, out of the different pathological sites, the key sites that have been found are lysine 274 and lysine 281. So our lab goal was like if we use a certain genetic model organism, which is C. elegans in our case, and we can introduce this kind of different PTMs in the C. elegans genome, what are the different pathological features that we can phenocopy using C. elegans as our model system? So since C. elegans doesn't have any endogenous tau by itself, so with the help of microinjection, we introduce first of all single copy of wild type human tau. As I said, like we use the 0 and 4R isoform. So with the help of microinjection technique, we introduce the single copy of that. And then with the help of CRISPR-Cas9 gene editing tool, we modify certain pathological sites. For example, threonine has been converted to glutamic acid to mimic phosphorylation. That is our phosphomimetic strain. And also correspondingly, lysine has been converted to glutamine at 274 and 281. That is our acetyl mutant, acetyl mimicking mutant. And we have a corresponding negative control for phosphorylation where threonine has been converted to alanine, which is basically a phosphoablation or phosphonol. So to summarize, the main C. elegans strains that are running in our lab, first of all, is just the wild type worms, which doesn't have any exogenous tau. Then we have the wild type copy, single copy of the human tau, that is the tau T4. And these are our two main pathological mutants. One is mimicking phosphorylation and the other one is mimicking acetylation. And as I said, we have a corresponding negative control. Couple of other things that I like to mention. We didn't want any whole body expression of tau because we wanted to limit the expression of tau. So neurons, these are the mechanosensory neurons in C. elegans because they have the longest microtubule structure. So we wanted to focus our tau solely expressed in only these neurons, not overall whole body organism level. And that we trace it with the help of a fluorescent dye called DENDRA2, which helps in tau localization as well as reporting tau turnover. But we have seen by conducting independent expression analysis, like DENDRA2 doesn't cause any pathological phenotypes. So just a basic background for the C. elegans as a model system. So it is a very short 1 millimeter size nematode species. It's very easy to maintain and very economic, and it develops quickly. Like this whole life cycle gets completed within 3 days. Like it has starting from the embryo, it has 4 larval stages, L1, L2, L3, L4, and then it moves to the adult phase. And this whole thing can be completed within less than 3 days. And also it's a great model organism to do aging studies. And I like to mention, since we are working on Alzheimer's disease, we do all our experiments in aged worms, which is like L4 plus 3 days. And then we age the worms 1 week later, and we conduct one more time at L4 plus 10 days. And also it has a very simple nervous system consisting of 302 neurons and 50 glia-like cells. And the advantage of our system, like our model system that we have homemade in our lab, is a couple of reasons. First of all, as I said, like it's a single copy transgene. Like you can ask me like whether there are any other available tau strains in the C. elegans field or not. My answer will be yes. But the problem with those, like they're like multiple copies and those are whole body expression. And when you have both of them, it causes the worms to be very sick, and obviously you cannot phenocopy any experiments. That's why we wanted to limit the expression only the mechanosensory neurons, and that too by using a single copy model. So these are basically our advantages compared to the rest of the available tau C. elegans model. So once we have all the strains optimized in the lab, we move on to the phenotypic characterization. For example, at the organism level, we check the neurodegeneration, we check the lifespan, we check the behavior. And as well, when we go more intracellularly, we check the mitochondria or any other cellular parameters that can be equally affected by this pathological mutant expression. So to start with the different results, first I will like to show my neurodegeneration assay, because since we are working with a neurological disorder, the first experiment, the key experiment that comes in our mind, like when we have these pathological mutants expressed, whether they cause any kind of neurodegeneration or not. So for that, we use a mechanosensory M-cherry reporter, which specifically expresses M-cherry in the mechanosensory neurons, and we cross it with all our tau strains that I listed before. And once the worms are already optimized, we place them in a glass slide with some anesthetic in them so that they don't move around. And then we move into the confocal for image analysis. So just a little background for this mechanosensory neurons, like there are 2 pairs of main mechanosensory neurons that are responsible for sensing light touch. So they like they grow from the head, like this is the head portion of the worm, and they go till the mid-ventral position. And then there is the other set of neuron that starts from the tail and it also ends in the mid-ventral position. The key thing to note, like these two neurons, they don't overlap with each other, so that the worms has a distinct anterior mechanosensory field and this is a distinct posterior mechanosensory field. And interestingly, when we did the assay at day 3 of the worms, as you can expect in the wild-type reporter strain, there is no overlap between the two neurons. But our phosphomutant, which is also in human, shows early disease phenotype. Here, the first alteration of the neural morphology that we see is the crossover of the two neurons. And then we quantified over a cohort of worms how many of those worms are showing this overextension defect. And as you can see, phosphorylation clearly has a 40% this defect as compared to the rest of the worms. So as I said, like we do our experiments like both at day 3 and day 10. So same worms, we aged them 1 week later. And here, in addition of the overextension phenotype that we have seen in the phosphomutant, we see other cohort or different defects, as I will show you. So first we will see the branching phenotype in the same mechanosensory neurons. And also we see misguidance. Basically, these neurons are like long linear in structure, but here we can see a wavy phenotype happening. Also, we see a bead or pearl-like structure that happens in the neurons. Then there is a breakage within the neurons as well, which can eventually lead to complete neuronal loss. And in severe cases, we also can see multiple neurodegeneration phenotype in the same single neuron. So most of these, all these different neurological defects, we can see only in the phospho or the acetyl mutant, which we have quantified in the next slide. So as you can see, all these tall graphs, the red indicates the phospho, the green indicates the acetyl. They're significantly more higher compared to the rest of the control worms. And I like to pay your attention, like even the wild type tau, they have like very severe, like very less amount of percentage of defect. Only the modified tau mutants, they are severely affected compared to the other controls. And even the phosphoablation is behaving very much similar to the wild type. So this says like all the tau mutant strains which are modified, be it phosphorylated or be it acetylated, those are the more affected compared to the rest of the control group. Now, since we see a morphological defect, the question comes like, does it hampers the function of the neurons or not? So as I briefly mentioned, like these neurons are mainly responsible for sensing light touch. So we also do that assay by taking like an eyelash, or even you can take a skin hair and gently touch the worm head. So it will elicit a backward response, as you can see in this video. So interestingly, here also, when we quantify like how many worms are able to sense that light touch, we see both the phospho as well as the acetyl mutant, they are severely light touch insensitive, indicating that their morphological features were affected. Simultaneously, their function was affected as well. And also here you can see at day 3 only the phosphorylation mutant was affected, not the acetyl mutant. But as we age the worms, then both the mutants show this kind of defective phenotype. And other thing I like to mention, like as I mentioned, like this precise mutation was solely expressed in the mechanosensory neurons. We wanted to just make sure like whether any other kind of behavioral affected behavior are affected in the worms, in this set of worms or not, which are mediated by other neurons. For example, thrashing, which is like a swimming behavior of the worms. We quantify how many body bends or thrashes it made in the liquid, as well as the normal locomotion on the solid agar surface. We count the how many body bends the worms made over a certain period of time. And interestingly, here you can see like none of the strains are like different from each other, indicating like since our tau is expressed in those sensory neurons, that's why those are affected. But other behaviors which are mediated by other kind of neurons, those are still working perfectly fine. And also we check the overall lifespan of the strains. We didn't see any kind of lifespan difference across the different strains. Okay, to summarize this portion, we can say in our lab, we have an established working model where we can produce subtle phenotypes which are progressive with age. and which are restricted to the pathological mutants. Like even the wild type tau mutant, those are acting perfectly fine, but only the modified tau strains, we can see the pathological behaviors or the neuronal abnormalities in those mutants only. So now the question comes, okay, this is more on the organism level, but what happens exactly within the cellular level? And since we are talking so much about the neurons, the first key cellular organ that comes in our mind is mitochondria, because as we know from our high school studies, like neurons are rich in mitochondria and they provide ATP for the neuron to carry out different functions, for example, transport, synaptic transmission, etc. So our main cellular interest then becomes the mitochondria. And over the years, we have seen such fantastic studies from Professor Dr. Albensi, Dr. Rady, Dr. Perry, Dr. Elegan, like who are the pioneers in this field. And in their various research studies, they have shown like when there's a mitochondrial dysfunction, whether due to aging or due to unhealthy diet, it alters all the different parameters of mitochondria, for example, morphology, transport, ATP generation, etc., which further aids in the progression of Alzheimer's disease. So our novelty, or I would say like our main interest was like when we have this modified tau, whether it causes similar kind of a dysfunctional mitochondria, which can be the backbone behind the neuronal deficits. And to check that, to check the mitochondrial different parameters, we use a specific fluorescent probe called MITO-M-chimer. So basically it's a pH dependent fluorescent probe, like it changes its color depending upon the pH of the medium. For example, if the pH is over 7, it will give you green color, whereas if the pH drops below 5, it will give you red color. And it is specifically targeted to the mitochondrial matrix itself. And also we can analyze at 2 different wavelengths. Like for example, 440 wavelength will be your excitation. And then we have the 550 wavelength, which will give you the green channel, whereas the emission wavelength remains consistent at 550. Can you guys hear me?
Yes, we can hear you.
Okay, I think it got stuck for a minute. Okay, so as I was saying, like, so with this help of specific protein, we can analyze like different parameters of mitochondria. For example, in the dual mode, we can check mitophagy when we have the both green and the red channel simultaneously. Then using only the green channel, we can check the morphology features of the mitochondria. And then only with the red channel, we can check different trafficking behavior as well. So here, just a simple video. This is the cell body of the neuron, and you can see the mitochondria, they're moving back and forth along the neurite processes. So the beauty of this fluorescent probe, like at different wavelength or different channel, you can analyze whatever parameters you want to analyze. Okay, so like as always, once we got the reporter strain running, we again crossed back to all our tau strains, and then we moved on to characterize different parameters. So the first experiment that we did was the morphology analysis. So as we know, like in normal baseline scenario, mitochondria exist in this tubular reticular format, but with age or with other exogenous stresses, it causes aggregate formation or more like a punctate formation, punctate mitochondria. So in our case, we classified that into 3 different classes. Like if we are seeing one single green blob, we call it class 1. If we see more than 1, like 2, we call it class 2, or if it's more than 2, it is class 3. Then again, like always, like over a certain population, we quantify how many of the worms is exhibiting this kind of classes. And here also only at day 10, here I should mention, like we didn't see any difference at day 3, but only at day 10, we see both the acetyl and the phosphomutant, they are mostly demonstrating all the class 3, more fragmented mitochondria. So as we know, like this dynamic morphology of mitochondria is maintained by 2 dynamic processes, fission and fusion. There are a lot of different proteins involved, like DRP1, FISH1, all the mitofusions. So the beauty of the C. elegans model system, like we already have all the different mutants in our lab running. So at any point of time, if you want to dissect more on the mechanistic aspect, like what are the proteins that are being affected due to these tau modifications, we can easily cross it with our different C. elegans mutant strains. And that is one of the things that we're planning to do in future. So the next interesting mitochondrial mechanism, quality control mechanism that we will look for is the mitophagy. And as we all know, when normally we indicate mitochondria, like we see the mitochondria in the green channel, but as soon as it starts like accumulating damages, it changes its color, it becomes more acidic pH and it converts to red color. And then it gets eventually engulfed by mitophagosome, and which gets engulfed by lysosome, and it eventually degrades within the lysosome with the formation of mitolysosome. So this whole dynamic mechanism we can easily see in our C. elegans model system with the help of SEM fluorescent probe M-Primer. So for that, we take the image of the same cell body. As you can see, this is the green channel and this is the corresponding red channel. And then we do a quantitative analysis, more like a ratiometric analysis between the red and the green channel. Interestingly, at day 10, we didn't see any decrease in mitophagy across the different mutants. You can see like a slight little drop in mitophagy in the phosphor the acetyl mutant, but it was not statistically significant. So this is not the end of the story. Then we moved on to follow where those damaged mitolysosomes go, like whether they are properly trafficked across the neuron or they are more centrally localized. And for that, we did the transport or the trafficking experiment as well. So hopefully you can watch in this video because sometimes it's a little difficult to follow. So this is the control ablation strain, the tau strain. So as you can see, the white dots, it clearly moves back and forth across the neurite processes. So the videos are taken in the confocal and then we transfer them into the software called IMARIS, and then we quantify all the different dynamic movement of these mitolysosomal particles across the neurite structure. So this was in the control worm, but check what happens in the pathogenic tau mutants. It seems like mitolysosomes are present, no doubt. There is no decrease in the number of mitolysosomes, but it seems like they're stalled, like there's no movement across the neurite, like they're very stationary. So this is one of the key phenotype that we have discovered, like in the control worms, all the mitolysosomes, they're moving, they're having proper retrograde transport across the neurite process. But somehow in our pathological mutants, there seems to be like very stationary. And then with the help of ImageJ, we quantified all the different parameters. For example, as I said, we quantified the number of dynamic particles that were moving. And as you can see, both the phospho and the acetyl mutants, they are significantly less dynamic compared to the rest of the controls. Then we quantified the total run length, the distance of each and every particle, as well as the speed of each and every particle. And in both the cases, both the panel B and C, you can see the phospho and the acetyl mutants, they're significantly less dynamic compared to the rest of the controls. We even plotted chymograph plots where the zigzag lines indicates how dynamic those particles are. Whereas as expected in the tau mutant, phosphorylation tau mutant, you see like they're all straight line indicating they're very stationary. So this is one of the projects that we are currently right now working on. And we want to know the mechanism behind this, like why only in the pathological mutants we see like this kind of stationary mitolysosomes. There can be multiple reasons. First of all, like there can be less ATP, like maybe those neurons don't produce enough ATP for the particles to move across the neurite processes. And for that, we are developing another fluorescent probe called ATIM, which will basically indicate the ATP level inside those neurons. Also, since we are talking so much about the neurite, there will be a question like, what about the microbial structure and morphology in those specific worms? So we are working on that as well. And also, as we know, like whole this trafficking movement is carried out by different motor proteins, as well as mitochondrial proteins like kinesin, dynesin, etc. And again, like, as I said before, like C. elegans model system has all those different orthologs, and we have all those strains we can easily work out and find out the mechanistic aspect behind the less stationary movement in the phosphorylation or acetylation mutants. Another thing that we have seen so far, but we have not pursued or not put into our first paper is like whether these neurons they form any kind of aggregates or not. So this is one of the control strain, the ablation strain. As you can see, the cell body is very clean. There is no aggregate formation in and or around the cell body. But interestingly, you can see in the phospho or the acetyl mutants, you see certain aggregates present around the cell body. And more in the acetyl mutant, it seems like they're moving like around the axon initiation segment, and they're much more bigger in size. We didn't do any detailed quantification. We just checked how many worms are throwing this kind of aggregates. And as you can see, both the phospho and the acetyl mutant, they are significantly showing this kind of aggregates present in the cell body. So that can be like causing like a roadblock for all the mitolysosomes to not move across the neurite process. We don't know that we have to work on more. But interestingly, this phenotype has been already captured in the mammalian system. Like as I mentioned, like some of the pioneer scientists like Dr. Meldenkau or Legan, they have shown like when there's a modification of the tau protein, like they cannot bind to the microtubules properly, and they generally float around. And that's why they cause like a roadblock and gets accumulated in the axon initiation segment, and which can eventually causes trafficking disorder or like any kind of synaptic loss, and finally functional impairment of the neurons. So it's good to see like we are actually phenocopy those kind of pathological mutants in our C. elegans model system, but we need to work out more on the mechanistic approach. Okay, so to summarize so far, what we have seen in our phosphorylation tau mutants, we see reduced mitophagy, we see decreased mitolysosomal trafficking, which can be due to the aggregate formation, but we don't know as of yet. But all this can lead to neuronal deficits, be it functional decline or morphological defects, which eventually can lead to disease onset. So just a couple of few more data I like to share. Like all these experiments were done at baseline condition, like there was no exogenous stress present, or the worms were not treated with any kind of oxidant. But if you remember only the mitophagy experiment, we didn't see any kind of significant difference. So what we thought, like what happens when we stress the worms with any kind of mitochondrial oxidant? So for that, we use the well-known parapet, which basically blocks complex 1, causing decrease of ATP and increase of higher production of ROS. So we just did one single experiment using parapet. And interestingly enough, as you can expect, like the mitophagy should ramp up when you treat them with any kind of oxidant, because the cells want to get rid of any damaged or dysfunctional mitochondria. But at both day 3 and day 10, it seems like the phospho or the acetyl mutant, the mitophagy cannot ramp up. It seems like basically it's completely impaired only in the stress scenario. So this was kind of surprising result for us, like why, as you expect, like the mitophagy should ramp up, but only in the stress scenario, the pathological mutants doesn't, like they lack the ability of increasing the higher amount of mitophagy. And interestingly, it has been shown in the mammalian model system, like they say, like there's a phenomenon called system overload, like when the human brain is already suffering from so much exogenous stress, and which can like eventually lead to higher dysfunction of mitochondria, which causes more fragmentation and less mitophagy to happen. And that is one of the major clinical symptoms. So maybe something like that we are seeing in our model system as well, like maybe all the bad mitochondria, they're just getting accumulated, they're not getting rid of. So that also we need to focus for future as well. And one of the reviewer question was like, what happens to the autophagy? Like, okay, you are seeing a mitophagy defect, but what happens to the autophagy per se, like the overall bulk autophagy? So for that, we used another reporter strain. As we know, like LC3 is one of the classical autophagy marker. So in C. elegans, we have the LGG1, which is the ortholog of LC3. So that we fused with both GFP and mCherry to indicate autophagosome as well as autolysosome. And then we quantified the amount of autophagosome as well as autolysosome across the different mutants at both the days. And we didn't see any kind of difference in the baseline condition. So the next question was like, what happened to the stress in the stress scenario, because only mitophagy was affected in the stress condition. So for that, to induce stress in the worms in terms of autophagic stress, we basically starved the worms, which is basically nutrient depletion. We didn't give them food for 8 hours, and then we quantified the same thing. And as you can see, like the number of autolysosome increase tremendously when there is a fasting scenario. There's the corresponding images that we can see. And interestingly, both in the acetyl and the phosphorylation mutant, we see the autophagy mechanism like happening perfectly fine in the fasting condition as well. Like there is no defect in the autophagy. So which can help us to conclude like whatever defects we are seeing is specifically mitochondria specific, but not in general bulk autophagy is being affected when we give them certain kind of stress. Okay, so some of the key points that I have shown or shared with you is like when we have this single copy of modified tau, it causes baseline defects in neuronal function. It alters morphology of the neurons. It causes higher fragmentation of in the mitochondria, and it causes defect in the transport of the mitolysosomes. And in terms of mitophagy, only the stress-induced mitophagy is affected, but not autophagy. So all this data, or 80% of the data, was accepted or published in a couple of articles very recently. And also I will be giving a talk in the cell biology symposium in December. So since we have few more time, I will just quickly go through like what are the other experiments that we're conducting in the lab. So one of the very interesting system that we are working around now is the auxin inducible Degron system. So basically this is like auxin is like a phytohormone or the indole acetic acid, and the Degron is like a short motif of 44 amino acids in length. So the whole mechanism is like when your protein of interest is fused with this Degron system, and then you introduce in a system which has the receptor of it. And when you have the, in addition, you treat them with any auxin system. So this whole system identifies your protein of interest and targets it to proteasome and degrades it. So the main function or the main thing that we want to achieve, like if we think like the modified tau is the backbone of causing all these pathological phenotypes, then if we remove the tau system from the body, whether the worms will go back to their normal baseline level or not. So that is the main function for doing this kind of experiment. Like we tag them with this Degron's motif. Hopefully it will degrade in the proteasome with the presence of auxin, and we can revert back all the pathological phenotypes. Some of the key points or advantages, as I mentioned, like in C. elegans field, we have a huge C. elegans bank where we can easily order different promoters which can drive tissue specific expression. So not only the mechanosensory neurons we are working with, in future, if we want, we can work with other neurons as well. That is also one of the idea. And also this Degron tag can be efficiently introduced by CRISPR-Cas9 that we are currently working right now. And the other important thing that we can see, like when we remove the tau thing, how quickly it gets back. Like first of all, whether it gets back to the normal level or not, and then how quickly it can go back to the normal level after it gets cleared. So this is one thing that we are currently working on right now. And for a future project that we can think of, like since C. elegans is such a very simple tool to work with, like it will give us idea of working, what are the features that we can look in the higher model organism. So at some point we like to do a drug screening, which can like take more like a combinatorial approach. Like first of all, some of the drugs, if those can clear the tau aggregates, and the other ones can improve the mitochondrial health, and then we can do a cocktail approach and maybe work on primary neuron culture, and eventually if it works successful, we can even move into the higher model organism. So that is more like a PhD project that we can start after we get the answers for some of the mechanisms. So basically that's it. With that, I like to thank my two labs I'm part of. So this is Keith Narke, one of my mentor who works primarily in C. elegans, and this is his lab, and this is Dr. Gail Johnson, who mainly works on the tau field. And very recently, we got a R1 grant, which will help us to conduct the research more for the next coming years. And at the end, I'd like to thank you, Dr. Albensi, for giving me the opportunity to talk here. And also thank you for your attention, and we'll be happy to take any questions.
Okay, that was really great. Thanks for presenting that. I just have a couple quick general questions, and then I'm going to open it up to the audience for their questions. We don't work with the C. elegans model in my lab, although I do have a postdoctoral fellow, Dr. Ed Limagadam, that has. So what can you tell us about C. elegans models regarding tau that you can use to your advantage that we can't in mouse models?
Yeah, so one of the biggest advantage is like the time frame, because as I said, like the whole life cycle of C. elegans completed within 4 weeks. So all these pathological phenotypes that we were trying to phenocopy, we can like do a time course analysis. We can progressively monitor them. For example, when we do the assay at day 3, the same worms, we can conduct them like after 1 week, whether those due to aging, whether those phenotypes exacerbates or not. So for example, the neurodegeneration experiment, right? We see certain kinds of phenotypes only at day 3 with the phosphorylation mutant. But as soon as we age the worms for another week, we can see those same neurodegeneration phenotypes in the acetyl mutant. So in terms of timeline, very quickly we can assay those experiments, whereas in mouse model, I think it should take at least 8 to 10 weeks. That one of the major reason. And also the other reason is that the C. elegans has a transparent body. So all these fluorescent reporters or the fluorescent probes, we can easily integrate in their genome and just do live imaging. Like for example, the traffic experiment or the morphology experiment, we can easily conduct all this live imaging very easily and promptly as well using this model system. So I think, for example, if you want to model a disease, it's very easy to start with C. elegans, and then as you move into the higher model organism, you already know what to look for. And then you quickly work on those specific assays instead of just fishing around.
So you had several interesting observations, including changes or effects with mitolysosomes. Mm-hmm. And you showed a video where the mitochondria are being stalled as they move around. How much of this is generalizable to other neurological disorders? I wasn't quite clear on if you were talking about a specific disease condition or not in that example. I might have missed it.
Yeah, so it's mostly related to, I would say, more like tauopathy disease, which is the bigger umbrella of all this PSP, AD, and all those. So specifically in the tauopathy field, what they're saying, like since tau is the main protein to maintain microtubule assembly and their organization and structure, so as soon as it gets modified, its structure changes, right? And then it cannot bind to the microtubule properly, and it generally floats around. And that's how the microtubule starts degrading. And since all these mitolysosomes or mitochondria per se, they need to be trafficked across the neuron. And if there is a microtubule disorganization, then there will be a trafficking problem as well, because they don't have the proper parameter to travel across the neurite processes. So I think it's more to the tauopathy related diseases, but not sure. Like for example, if you ask me whether it happens in the Parkinson's disease, my answer or my guess will be probably no.
Okay, so let's open it up to the general audience. And I do see a couple of questions in the chat box. I see one from Ida in my lab. Ida, I don't know if you want to just unmute and ask the question, but she's asking about whether or not you use hermaphrodites or male worms, and if you injected tau into the worm's gonad. So I don't know if you can read her chat comments or not.
Yeah, so that's a good question. So all our experiments are conducted only in the hermaphrodite worms. When we create a new transgenic strain, then we need the male for mating. But in terms of experimental analysis, we used all hermaphrodites, and yes, they're directly injected into the C. elegans gonad. That's correct.
Okay. Other questions?
I have a question. Hi, I'm Pranav. Can you hear me?
Yeah.
So there was this video that you showed wherein touching the head of C. elegans made it to move backwards. So how exactly can we relate that experiment to Alzheimer's disease or neurodegeneration? If you could just throw some light on that. Thank you.
Yeah, so that was more like a phenotypic readout for our assay because as I mentioned very briefly, I think I should have explained it more. So like in the C. elegans system, there are huge class of mechanosensory neurons. And out of those, we are only focusing only on those mechanosensory neurons which are responsible for light touch, not all the mechanosensory neurons. And that's why we introduce our tau in those specific light touch mechanosensory neurons. So our main goal for that experiment is like when you have this pathogenic tau, whether those neurons which are responsible for sensing light touch, that gets hampered or not. So it's not more like a disease perspective, it's more like whether our model system is acting fine or not. Because, and that's why I did other bunch of assays, like for example, thrashing, locomotion, which are regulated by other neurons, and we didn't see any kind of defect in those cases. So that will indicate like only the light touch mechanosensory neurons are affected, but not rest of the other neurons.
Okay, thank you.
Yeah.
Other questions from the audience?
Yeah,
I
so
think I
someone,
see
yeah.
one here from Raj. How much can we relate C. elegans to in vivo results from Raj?
Yeah, so if you think like this is also like more in vivo because we are working with the whole organism level, right? It's not like we are doing any cell culture in vitro work. It's more whole body C. elegans experiment that we are doing, and we are doing all the neurodegeneration assays, all the behavioral assays and everything. So they are very much relatable. And of course, like when we think of the human disease perspective, there is a huge leap, right? And we have to do step by step. In those cases, I think maybe mouse model will be more helpful. But since we know what to look for, even if we work with a higher model organism, we can definitely go for those assays. But as of now, I think for just phenocopying a disease perspective, C. elegans, I think, is a great tool for us to use.
Okay, that's great. Any other comments?
Hi there. I do have one question. Hi there.
Yeah, hi.
Very impressive set of data. You had mentioned that the C. elegans does not express tau endogenously. I was wondering if they express A beta endogenously.
A beta, yes, A beta, I think so, but the exact homology or with the human protein is not 100%. I think they're less than 50%. And in case of tau also, I should mention like there is a protein called PTL1, which stands for protein tau-like. It's also, it shares like, I think 20-25% with the human tau. So it's not like totally 100% or totally 0%, but both of these proteins, like they have a very minute sequence similarity in the C. elegans model system, but not like a over 80% match or anything like that.
Okay, thank you.
Aggregates inside the lysosomes from your experiments?
I think there is another question or comment, but I don't think most of us could hear it. Could you try again?
Hi, I'm Narendran here. Hi, Sanjay. Did you able to observe tau aggregates inside the lysosomes with this phosphorylation mutant?
Yeah, that's a good point. Yeah, so in one of my slide, I have mentioned like all our tau is tagged with the protein called dendro2. That will actually help us to see or localize of the tau specifically inside the cellular organism. So that is one of the things that we are currently working on to see tau localization as well as tau turnover over time. And that dendro2 will be help us for doing that, that kind of experiment. Like exactly where it is accumulated or whether it is getting degraded over time or not.
Okay, great. So I don't know if there are any final questions there now. We certainly have a couple more minutes. I'll just give people a chance to ask one or two more questions, but until we see those pop up in the chat, I'll just remind everyone we're going to do this again next month. Next month, we have Dr. Brian Balin from the Osteopathic College of Philadelphia speaking. Right now, I think it's in general terms, but I'm not sure, on infectious disease and Alzheimer's disease. And so he'll be presenting that sometime in mid-March, and we'll use the virtual format again. And so I just want to remind everyone, we attempted to record today's session. We'll see if that worked out. And I'm also going to post the two recent publications that, that Dr. Guha just published in regarding some of this data. I'll present that and post that on our Manitoba Dementia website, and that link will be found on my LinkedIn profile. So if there aren't any final questions, thank you all for.
Question from Ida.
Okay, good. Good.
Yeah,
go ahead, Ida. Sorry.
Yeah. So yeah, as I mentioned, like whatever other C. elegans model system of tau is present is like they're already having the overexpression of tau, and it's more like a whole body of tau. So those kind of worms, like they're very sick, like they hardly can make 2 weeks. So when we definitely use them as our control with our strains, and you saw my lifespan data, like since our worms just has the single copy of tau, and that too expressed in certain set of neurons, it didn't cause any lifespan defect, but those worms died within 2 weeks. So those are very sick, where our worms were happy and healthy otherwise.
Okay, great. Thanks, Ida. Anyone else? Okay, well, thank you again, Dr. Guha, for presenting today. I really appreciate your time, and also thank your mentor for us for allowing you to give this presentation today. And I hope everyone that's attending has a chance to join us next month for our next Dementia Journal Club. So I'm going to sign off now. Thank you very much.
Thank you. Thank you.
Okay, bye-bye now.
Bye.